Welcome to All Force e-learning. Now today we are going to discuss that is differentiation of Mesoderm. Now already in the previous class we have discussed about gastrulation process. During the gastrulation process itself, what happened here is the formation of three germ layers, notochord as well as nerve cord. Now this is what the diagram shows. Here is this is that ectoderm, this is that endoderm, now this is that mesoderm, this is the nerve cord, this is the notochord. Now these are the three germ layers along with nerve cord and notochord is formed during the gastrulation process. Now how this mesoderm is differentiated into, what are the different parts the mesoderm is differentiated into. Now if you observe the mesoderm, here is this is the ectoderm, this is the endoderm. Now in between this one is what we are saying mesoderm. The mesoderm is differentiated into three parts. Now the mesoderm, the part of the mesoderm which is present just besides the nerve cord, this part, this is called as epimere or otherwise also called as paraxial mesoderm, paraxial mesoderm. The mesoderm present just besides the nerve cord and notochord. This is the paraxial mesoderm which is present in the form of a longitudinal axis of the notochord. This is the paraxial mesoderm or epimere. Now the part of the mesoderm which is surrounding the gut, now this is what we call it as hypomere. Now it is also called as lateral plate mesoderm, lateral plate mesoderm, this is also called as hypomere. Then the middle mesoderm, this part of the mesoderm is mesomere, this is that mesomere, this is. Now mesomere is also called as intermediate mesoderm. Now what we are discussing here is mesoderm differentiation. How the mesoderm is differentiated into. This is that mesoderm, this is the ectoderm, this is endoderm, this is a nerve cord, this is notochord. In between ectoderm and endoderm, the mesoderm is represented in the form of, this is three parts. The part of the mesoderm which is present on either side of the nerve cord, this is what we are saying epimere in the form of a longitudinal axis. Now the part of the mesoderm which is surrounding the gut, now this part is what we are saying hypomere or otherwise lateral plate mesoderm. Now the part of the mesoderm in between the epimere and hypomere, this is mesomere or intermediate mesoderm. Now this is the position of mesoderm and parts of the mesoderm. Now this epimere, mesomere, hypomere, these are the three parts that differentiated into, the mesoderm is differentiated into these three parts. Now what are those which arises from the epimere, mesomere and hypomere? What are the different areas and what are the different parts or organs which are obtained from these parts such as epimere, mesomere and hypomere? Now if you observe the hypomere, this hypomere is split into two. This is outer, this is inner. Now outer, this is outer is what we are saying somatic mesoderm. This is somatic mesoderm. Now inner splanchnic. Now in between these two there is a cavity. This cavity is called as coelom. Now it means hypomere, this is that hypomere, it splits into two parts, that is outer somatic mesoderm, inner splanchnic mesoderm. In between these two there is a cavity, that cavity is called as coelom. Okay. Now what are the different parts which are arised from this mesoderm? Now we will discuss here what are the different parts which are formed from this mesoderm. Here is the mesoderm which gives rise to the following parts. Now here is, this is mesoderm. Now it is divided into these following three parts. We are saying this is epimere, adjacent to the notochord or nerve cord, epimere. Then there is mesomere, this is middle. Then there is hypomere. Now epimere, what it gives rise to? Epimere gives rise to, now epimere is again divided into blocks. Now cubical blocks. The epimere is divided into cubical blocks on either side. Now these blocks, this epimere is divided into cubical blocks. Now they are three cubical blocks, that is sclerotome, myotome, dermatome. Now these are the three cubical blocks which is divided into, the epimere is divided into three cubical blocks: sclerotome, myotome, dermatome. Now if you observe in diagrammatic view, now this is how it looks like. Now this is that part what we are showing here. This is the primitive node forming primitive pit. Now this is that part, primitive groove. Now on either side it has blocks. It has blocks. Now these are the blocks of mesoderm. Now each block of mesoderm is divided into three. The three parts: sclerotome, myotome, dermatome. Now this is the blocks. Each block is divided into three parts, that is sclerotome, myotome, dermatome. Now sclerotome, it gives rise to, the sclerotome gives rise to, what is the part which it gives rise to? It gives rise to skeleton. Whereas myotome, it forms muscles, the voluntary muscles which are voluntary muscles. Dermatome, it gives rise to skin and connective tissue. Now these are the three parts which are obtained from sclerotome, gives rise to skeleton, whereas myotome gives rise to muscles, dermatome gives rise to skin and connective tissue. These are the cubical blocks which consist of one block, three blocks in each: sclerotome, myotome, dermatome. Sclerotome gives rise to skeleton, myotome gives rise to muscles, dermatome gives rise to skin and connective tissue. This is about epimere, which is present adjacent to notochord or nerve cord. Now mesomere, the middle part of, now it gives rise to urinogenital organs. Urinogenital organs means they gives rise to urinary system, that is excretory system, as well as gonadal system, reproductive system. It means it provides two. What are those two? One is excretory system. The other one is the gonadal system, that is reproductive system. Now these are the two which gives rise to, which one? Mesomere, urinogenital organs, excretory system and reproductive system or reproductive organs. Then hypomere gives rise to what? What is the structure which is gives rise to? Hypomere gives rise to. Now hypomere, it is divided into inner splanchnic, outer somatic mesoderm. Now this hypomere, it provides here, this is a cavity called as coelom. Now this coelom cavity, it provides peritoneal lining, pericardial lining, pleura. This is it provides peritoneal layer, pleura, pericardial layer, etc. Now this is the cavity which forms a coelom in between the splanchnic and somatic mesoderm. Now these are the parts into which the mesoderm is differentiated. The mesoderm is differentiated: epimere, mesomere, hypomere. Epimere into cubical blocks. These cubical blocks, 3: sclerotome, myotome, dermatome. Sclerotome, skeleton; myotome, muscles; dermatome, skin and connective tissue. Then mesomere provides urinogenital organs, excretory system, reproductive system. Then hypomere, it provides coelom, the body cavity, and peritoneal, pleura, pericardium, etc. These are the differentiation of mesoderm in humans. Then we have extra embryonic membranes. This is the topic here. Extra embryonic membranes. What are these extra embryonic membranes? Extra embryonic membranes are those membranes which covers the embryo during its development. During the development, the membranes which cover and protect the embryo, these are the extra embryonic membranes, also called as, sometimes referred as, fetal membranes. What are those? There are four in number. The extra embryonic membranes are 4 in number. What are their functions and what are their names and how they are formed? This is how they are formed. What is their function? Extra embryonic membranes. Here, this is the topic: extra embryonic membranes. Extra embryonic membranes. What are these extra embryonic membranes? Those membranes which surround the embryo or fetus during its development. During its development, those membranes which surround the fetus or embryo. Now embryo later forms into fetus. Now hence we are saying the membrane which is surrounding the embryo, hence we are saying embryonic membranes. Extra means as they are present outside. Extra embryonic membranes. What is their function? What is the function of this extra embryonic membrane? The function of this extra embryonic membranes is, first, providing nourishment. This is nutrition. Providing nourishment or nutrition to the developing embryo, as well as removes waste. Now oxygen, provides oxygen, remove carbon dioxide, provides oxygen and removes CO2, as well as does not allow the embryo to desiccate. Does not allow embryo to desiccate. It means not to dry up. The embryo does not dry up due to the presence of this membrane. Does not allow embryo to desiccate, as well as it also here another important provide here. This is mechanical shocks. Prevent from mechanical shocks. This is what we are saying, protection from mechanical shocks. Now these are the functions: providing nourishment, removes waste, provides O2, that is oxygen, and removes CO2, does not allow embryo to desiccate, does not dry up, as well as prevent from mechanical shocks. These are the functions of extra embryonic membranes. Now how they are formed, where they are formed? Now already if you observe, there are two membranes already formed, amniotic layer as well as yolk sac. Now how this is formed? If you observe here in the embryo, when there is bilaminar embryo is formed, when the bilaminar embryo is formed, we shown two structures here. What are these structures here in the bilaminar embryo? Here this is the bilaminar embryo. This is a cytotrophoblast. This is the cytotrophoblast. Now this is the syncytiotrophoblast. Then there are two layers of cells. One layer of cells is tall cells. Now these are the tall cells, one layer of cells. Now these are called as epiblast cells. We have said this is epiblast cells. They are arranged 2 layers. Okay. Now then another layer just below it. This is hypoblast. This is hypoblast. This is hypoblast cells. Now these hypoblast cells, they delaminate and forms here. This is a yolk sac. Okay. Then the epiblast cells, now towards their syncytiotrophoblasts cells, they produce amniotic cells and produce this structure. What is this structure? Is called as amniotic sac. This is amnion. This is amnion. Now these are the 2 layers, that is fetal membranes, which are already forming these ones. Whereas the part, what is this part which is present in between? This is cytotrophoblast and this is yolk sac. This part is what we are calling as, this empty space is called as extra embryonic mesoderm. This part is extra embryonic mesoderm. Extra embryonic mesoderm. Now this extra embryonic mesoderm, this part is what we are saying. Now the cells divide and redivide to occupy the entire part here, starting from the cytotrophoblast up to yolk sac. There are large number of cells which are formed here. These are occupied the entire extra embryonic coelom actually, which forms extra embryonic mesoderm. This cavity is called as extra embryonic coelom. Now that cavity is filled with, these are what we are saying, mesoderm. This is extra embryonic mesoderm. It means the cells in that cavity divides and redivides. Once again I repeat without confusion. Here the cavity between yolk sac and cytotrophoblast. The cavity between trophoblast, cytotrophoblast and yolk sac. What is that cavity? Extra embryonic coelom. Now this cavity is filled with what are these cells? Extra embryonic mesodermal cells. Now these extra embryonic mesodermal cells, a cavity appears, cavitation takes place. Now this cavity appears, now this entire cavity appears here, and this cavity which appears, it makes some of the cells migrate towards cytotrophoblast, some of the cells towards the yolk sac. Now these cells which move towards the cytotrophoblast, they form somatic mesoderm, whereas the membrane, this is which are attached to yolk sac or towards amnion, this is called as the inner side. They are called as splanchnic mesoderm. Now in the next diagram we are showing here, this is the part, it looks like this part, it looks like here. This is that cytotrophoblast. This is that cytotrophoblast. This is the cytotrophoblast. These are the, now this is the syncytiotrophoblaст. Okay. Now here there are cells, what we are saying, tall cells. Now here these are the tall cells. What are these tall cells? Are called as epiblast. Then there is a small cells in which are cuboidal. These are the cuboidal cells. These are the cuboidal cells. Okay, now this is formed here into yolk sac here. This is okay. Now if you observe above, there is which sac? This is amniotic sac. This is amniotic layer. This is okay. Now if you observe, these are the cells. What are these cells? Are called as, which are attached towards the embryo. These are called as splanchnic mesoderm. Now this mesoderm is extra embryonic. Now previously discussed mesoderm is present in this embryo, that is intra-embryonic mesoderm. What we have discussed earlier is intra. This is extra embryonic mesoderm. Now this extra embryonic mesoderm present towards the cytotrophoblast. This is cytotrophoblast. Now this is called as somatic. Now this is extra somatic. This is this is intrasomatic. Intra means here extrasomatic mesoderm. It is differentiated into two parts, outer, inner. This outer is called as, this is what we are saying, somatic. This is a somatic mesoderm. Now this inner one, it is called as, this inner one, this mesoderm, it is called as splanchnic mesoderm. Now these two are extrasplanchnic mesoderm. This is extrasomatic mesoderm. This is a extrasplanchnic mesoderm, because this coelom is what we are saying, this mesoderm is called as extra embryonic coelom. Now hence this is a extrasplanchnic mesoderm, this is extrasomatic mesoderm. Okay, now the somatic mesoderm along with cytotrophoblast, these two together form chorion. What is chorion? Chorion is formed by the fusion of, this is a somatic mesoderm along with cytotrophoblast, or otherwise cytotrophoblast also called as trophectoderm. Cytotrophoblast also called as trophectoderm. Now this chorion consists of one layer. This outer layer is called as trophectoderm. The inner layer is what we are saying, somatic mesoderm. Together we call it as chorion. Now similarly here, this is the inner, what we are saying, inner is splanchnic mesoderm along with endoderm. These two together form amnion. Okay. Now here this yolk sac is formed here, inner splanchnic mesoderm along with the endoderm, together it forms yolk sac. This is yolk sac. Now soon after trilaminar embryo is formed. This is trilaminar embryo. When it is formed, when we observe, this is a trilaminar embryo. This is third germ layer. This is the endoderm, mesoderm and ectoderm. Soon after the trilaminar embryo is formed, from the hind gut a small, what we are saying, sac-like structure arises from the hind gut. A small sac-like structure arises from the hind gut. Now this is what we are saying, allantois. Now this one along with the splanchnic mesoderm, together it forms allantois. Now remember one thing, that is the cavity occurs and some of the cells are migrated towards inside, some of the cells migrated towards outside, which are attached to the yolk sac. These are called as splanchnic mesoderm. These cells are called as somatic mesoderm. This cavity is called as extra embryonic coelom. This cavity is called as extra embryonic coelom. This cavity is called as extra embryonic coelom. This cavity is called as. Now what we are saying here is the entire cavitation has occurred and separated two layers, but here there is still connected, this embryo is connected to the trophectoderm by a stalk. This is what we are saying, a stalk with which the embryo is still connected. It means the cavity is present all over the embryo except at this region. Now this is what we are saying, the stalk, embryonic stalk this is. Now which later forms into what we call it as umbilical cord, because these cytotrophoblast cells, they form chorion. Now this chorion and allantois together form here, together it forms here chorioallantoic placenta. Now it means this is that embryonic stalk, this is STALK stalk, which is formed here. Once again I say here, I repeat here, what is, how actually the extra embryonic membranes are formed. First, this is the cavity. What is this cavity is called as extra embryonic coelom. That extra embryonic coelom, the cells divide and occupy the entire part here. Now this is called as extra embryonic mesoderm. A split occurs here. This is a cavity occurs, not actually split, a cavity occurs. This cavity makes the cells to move towards outside and inside. Now some of the cells attach it to the inside, some of the cells move towards outside. Now those cells of mesoderm which forms towards inside, this is splanchnic mesoderm, extra embryonic splanchnic mesoderm. Now these cells which are attached to cytotrophoblast, these are called as extra embryonic somatic mesoderm. Now this epiblast along with the extra embryonic mesoderm, this is what we are saying, somatic mesoderm, along with epiblast together form chorion. Then there is another one, this is the inner splanchnic along with this is what we are calling yolk sac, together form not yolk sac, sorry, amniotic cavity, the membrane endoderm, together form amnion. Then another one here is yolk sac, this is already yolk sac is formed. Now this is a splanchnic mesoderm attached to this, together form yolk sac. At the hind gut a sac moves out or comes out, now which get fused to the chorion in later stage. Now this is called as allantois, which has outer, this is splanchnic, inner endoderm, together this is called as allantois. This is the third membrane. Now these are the four membranes. What are those four membranes? First, amnion, chorion, allantois, yolk sac. Now we have already mentioned the functions. Amnion and chorion, if you observe these two, they together form placenta. Not amnion, sorry, this is a chorion and allantois together form placenta, exchange of gases, nutrients, etc. Now amnion, this is the amnion is that layer which actually protects the entire embryo by secreting fluid, amniotic fluid, into which the embryo floats, does not allow desiccation. This is amnion. Whereas yolk sac, we know that RBC are produced from the yolk sac during the embryonic stage, as well as in the 5th month some of the cells, gonadal cells, extra gonadal cells moves from the yolk sac. Now this is that yolk sac. Because yolk is absent in humans, because human development involves intrauterine development where it nourishes from the uterus. Hence, we observe yolk sac has no role in nourishment, as the yolk is absent or very less. This is. Now, if you observe here, these membranes, how they appear in one diagram, if you observe here, this is how it looks like, the membranes. The membranes look like this. Here is. This is the one what we are saying, cytotrophoblast. Now these are the syncytiotrophoblast. Now here this is the trilaminar embryo. This is. Then the entire embryo is covered by what is this structure is called as yolk sac. Now this is that yolk sac. Now which can not yolk sac, sorry, not yolk sac. This is this membrane is what we are saying, amnion, amniotic layer. Now here there will be allantois which fuses with the chorion. This is allantois which fuses with the chorion from the hind region. Now there is a yolk sac here, where the yolk sac is. The yolk sac get degenerated. Now this is that yolk sac. This is the yolk sac. Now this is allantois. Now this is amniotic fluid. Amniotic fluid. Now this is the amnion. This is the amnion. This is allantois. Now this is yolk sac. Now this structure of cytotrophoblast along with this structure is what we are saying now, chorion. Chorion. The trophectoderm along with the somatic mesoderm, extra embryonic somatic mesoderm together form chorion. This is outermost. This is allantois from the hindgut. The yolk sac which actually get degenerated with development. Then this is a amniotic sac which covers the entire embryo. Now this is, it is covering the entire embryo here. And hindgut, there is a sac like structure which is called as allantois. In humans, normally allantois has no function of excretion, because the placenta takes part in the process of excretion. Okay, now these are about extra embryonic membranes 4. Okay, thank you.